Enterobacteriaceae. Here are your objectives for this lecture. Enterobacteriaceae, most can be normal flora. Commonly isolated genera include the Escherichiae. Also, like when you hear E. coli, that's what the E stands for. Klebsiella, Enterobacter, Serratia, Proteus, Providencia, and Morganella. Salmonella and Shigella are always going to be pathogenic if recovered in a culture. Enterobacteriaceae are the leading cause of urinary tract infections, as well as bacteremia. These are gram-negative rods, which account for half of all clinically significant isolates found, and we need to perform antimicrobial susceptibility testing on all isolates. Enterobacteriaceae, again, they're all gram-negative rods. Oftentimes they're large, straight bacillus with rounded ends. Not all are motile, but if they are motile, they do so by having a paratrichous flagella. They are able to ferment sugars. They are found in the environment and the intestinal tract of humans and animals. A lot of times you'll hear this family referred to as enteric or enteric gram-negative rods. The enteric bacteria in water and milk are oftentimes due to fecal contamination. Additional general characteristics are that they have the ability to ferment carbohydrates, specifically glucose, the ability to reduce nitrates to nitrites, and they lack the oxidase enzyme, so they are oxidase negative. In the next unit, we'll move on to the non-glucose fermenters, so it's important to remember that those three characteristics are how you can separate Enterobacteriaceae from other gram-negative rods. So again, the ability to ferment carbohydrates, specifically glucose, the ability to reduce nitrates to nitrites, and they are all oxidase negative. Most will grow on commonly used media, typically within a day. Not too many of these are fastidious organisms. We can use differential media like McConkey's. If they're a lactose fermenter, we'll get a pink colony on that agar. If they are a non-fermenter, the colony will be clear or colorless. Our organisms can contain different types of antigens. So an O antigen is found on the cell wall, the K antigen is found in the capsule, and the H antigen is found in the flagella. So there are some of these organisms that are notorious for having a capsule. All right, first we will start with Escherichia, or commonly referred to as E. coli. They are lactose positive, so usually it is a normal flora in the intestinal tract of humans and animals. Most infections are going to be endogenous, so they originate from within the body. E. coli is a significant agent of urinary tract infections, diarrheal disease, sepsis, and neonatal meningitis. The babies that get that are usually exposed during childbirth. We now have enterohemorrhagic E. coli 0157H7. Several years ago, this was responsible for some deaths from undercooked hamburger that was contaminated with E. coli 0157. The individuals had bloody diarrhea or hemorrhagic colitis, and this can progress to hemolytic uremic syndrome or HUS. It's transmitted by ingestion of undercooked ground beef, can also be from raw milk and apple cider. The varotoxin is the major virulence factor that actually causes the disease to progress. Special augers are used, such as McConkey's with sorbitol, that is now routinely set for all stool cultures. So using McConkey with the sorbitol provides a selective medium that will allow for easier identification of E. coli 0157H7 by differentiating it from other lactose fermenting E. coli strains. Klebsiella, Enterobacter, and Serratia. So these are Vogus pascure or VP positive. The most important species are Klebsiella pneumonia. These are isolated from the respiratory tract and stool. Enterobacter aerogenes is down in the soil, dairy products, water, and sewage. And Serratia marcines is an environmental, so typically we will recover Serratia from things like a wound culture. So like the name implies, Klebsiella pneumonia causes bacterial pneumonia. Klebsiella is a large gelatinous capsule, and it will be a mucoid to the point where it could be like dripping from the lid. So they're pretty slimy, snotty, mucoid-looking organisms that are growing. The capsule is there to prevent phagocytosis, and it can also inhibit leukocyte migration. Klebsiella can be responsible for milder forms of respiratory infections, such as bronchitis and bronchopneumonia, and it can also come from nosocomial. Like mentioned earlier, Klebsiella pneumonia will present as a large mucoid-looking colony. They do ferment lactose, so you're going to get a pink color on your McConkey plate. They are non-motile. They are lysine, decarboxylase, and citrate positive, and they are indole negative. Enterobacteriaceae is motile. It also may have a capsule. It is also a lactose fermenter. It is citrase and orthonine decarboxylase positive and indole positive. And then Serratia marcescens is a highly pigmented organism. As you can see in the picture, it is usually a pink to orange kind of pigment. It is motile, and it is a slow lactose fermenter. We'll go ahead and shift to the next group of Proteus, Providencia, and Morganella. Again, these are normal flora of the intestinal tract. They are motile. They do ferment xylose. Common isolates are going to include Proteus mirabilis and Proteus vulgaris, Providencia retgeri, Providencia alficescens, Providencia stuarti, and Morganella morgani. Clinically significant of these organisms are your Proteus mirabilis. It's a common cause of urinary tract infections, and it is urease positive. So one way to separate Proteus from others is Proteus are known to be urease positive, while Morganella and Providencia are urease negative. With Proteus, these are considered lactase negative. You'll have a clear or colorless colony on McConkey. Urease positive, so you're going to have to set up a urea tube, or if you're looking at your Vitec card or your microscan panel, you'll see the biochemical in the urease is hot pink. Proteus, also is known for its swarming. So when you look at growth on a plate, you'll see waves or layers of growth across the entire plate. So if you look at the picture on the slide, kind of looks like a tree trunk, right? When you cut it down, you have like those layers like that. So the saying that you need to remember is if it swarms, it's Proteus, but not all Proteus swarm. So as you learn to read plates, if you see swarming on a lot of your plate, and it'll usually go clearly from one end of the auger to the other auger, it's considered a Proteus. But again, not all Proteus swarm. Proteus vulgaris and mirabilis differentiate by performing an indole. So vulgaris is positive while mirabilis is negative. So again, sometimes the way to remember that is positively vulgar. So if you just remember if the indole is positive, then it's vulgarius. Antimicrobial susceptibility is indicated on all isolates of gram-negative Enterobacteriaceae. Citrobacter is also present in the environment. It can be isolated from feces of humans and animals. This is generally an opportunistic organism. It can cause urinary tract infections as well as neonatal meningitis. And the most common isolates will be Citrobacter diversus and Citrobacter forendoi. Since Citrobacter is opportunistic, it will often cause infections in patients that have underlying diseases or those that are immunocompromised. It is citrate positive, it is motile, and it will ferment lactose slowly or maybe not at all. It's a weak urease positive as well. Citrobacter forendoi is often H2S or hydrogen sulfide positive, so that colony may appear with a black center, and then that little black dot is the H2S production. We're going to do susceptibility testing against the aminoglycosides, chloramphenicol, and tetracycline. Yersinia is a gram-negative rod, and it can exhibit bipolar staining. Animals are actually the natural host, so we are actually an accidental host. Yersinia can be transmitted to humans by an insect vector or ingestion of contaminated food and water. Yersinia pestis causes the bubonic plague. It's transmitted by the flea on the rodent, so it's not actually the rodent that causes it. It is actually just the flea. So if you think of a prairie dog that has the flea on it, that has Yersinia pestis, that's the flea is the one that's actually responsible for the bubonic plague. The bubonic plague, this was characterized by swollen lymph nodes. The most serious complications included meningitis and pneumonia. The pneumonic plague results from inhalation of respiratory droplets from an infected person or animal. It's highly contagious and often fatal if untreated. It can arise as a complication of bubonic plague or primary infection. So just a quick overview of the bubonic plague. This is the most common form of plague caused by Yersinia pestis. It is transmitted primarily through the bite of an infected flea that infests rodents. For clinical features, we'll have incubation period of typically 2 to 6 days after the flea bite. It is characterized by a sudden onset of fever and chills, headache, fatigue, and malaise. They'll have those swollen lymph nodes. It'll be painful, swollen, and tender lymph nodes, usually in the groin, axilla, or neck. The skin over those lymph nodes may become erythematous and may ulcerate if untreated. Can lead to serious complications. So if untreated, the infection can spread, causing septicemia, which is a blood infection, meningitis, which is infection of the meninges, and or pneumonia, which is secondary pneumonic plague. Transmission of secondary forms, so pneumonic plague can develop from bubonic plague through hematogenous spread. Pneumonic plague is contagious via respiratory droplets and can cause outbreaks. Yersinia will look like short, plump, non-motile rods. A definitive identification is using an immunofluorescent stain of a direct smear, as you can see in the picture. It does grow best at 30 degrees, so again, a little outside of what your normal incubator temp is going to be, for that body temp of 35 to 37. It is lactose negative, and sometimes with Yersinia, we'll actually do serology testing versus trying to grow the organism and doing a traditional culture. So if we have an antibody titer of 1 to 16 or greater, or if we actually are able to draw both an acute and convalescent serum, and we see a 4-fold increase in antibody titers between the two serums, then that's also indicative of a positive infection. As far as treatment for Yersinia, streptomycin is, at the time, highly effective. We're going to switch gears once again to stool pathogens, and we're going to talk about Salmonella and Shigella. Salmonella is a normal flora found in the intestinal tract of animals, and Shigella typhi is often present in humans. Salmonella is the most common cause of diarrhea due to ingestion in contaminated animal or animal products such as poultry, dried or frozen eggs, dairy products, shellfish, beef, and pork. So this is why, you say no raw chicken on a cutting board, if you're going to be cutting up salad and other items as well, because it could be contaminated with that raw chicken because it hasn't been cooked yet to kill off that salmonella. You can also find it in contaminated water with feces of infected animals and humans, also as a transmission mode for humans. As previously mentioned, Salmonella is always pathogenic. The enteric fever is the most severe clinical manifestation of the infection that's caused by Salmonella typhi. Salmonella typhi is typically not killed by phagocytic cells and actually are even able to replicate within the macrophage. Salmonella can cause bacteremia and can cause enterocolitis, formerly called gastroenteritis, and this is the most common result of a Salmonella infection. Salmonella typhimurium is the most common species in the United States, and this is a reportable disease to the health department, and they will be tracking that contamination to help try to prevent it from getting any worse. As far as culture characteristics go, Salmonella are lactose negative, so you will see a clear or colorless colony. Typically for stool cultures, again, think back to your specimen collection and your traditional culture methods lecture. We do stools a little bit differently than we do others, where we're always trying to rule out pathogens for a stool. If you think about it, there's a lot of normal flora in there. It's going to be difficult to separate, right? So we're going to set specific augers to try to enhance the recovery of Shigella. When we're setting a stool, we use an SS auger, which is Salmonella Shigella auger. And sometimes you'll see XLD or xylose lactate dehydrogenase, and HE, which is hectorin enteric auger. They enhance the growth of Salmonella and Shigella. So it'd be a differential auger because you get fermenters and non-fermenters as far as glucose or lactose is concerned. So E. coli, since it's normal flora, and a stool will grow on an XLD and an HE auger plate. You're going to see color. So your plate may turn yellow, the other plate, the colony morphologies may be orange. Where we need to be concerned is on the XLD and HE plates, where we have clear or colorless colonies, particularly if they have that black dot or black center. That's the H2S production, which again is indicative of Salmonella. And you're also looking for those colorless colonies as well. Salmonella typhi produces H2S, so therefore, again, your colonies are going to have that black center. If you look at the picture on the slide, you can see that black on there. It's not always that prominent, but it can be. Other times it is like a legit little black dot in the colony. So if you're looking at an HP or an XLD plate and you're seeing clear or colorless colonies with a black center, you'll need to think Salmonella. Organisms are usually recovered from a stool, but they can also be found in blood. And oftentimes, Salmonella infections are self-limiting, so you may feel like you had food poisoning for a few days and then it will resolve on its own, but it can be more serious. And if neonates do get this infection, they will require treatment. Shigella has highly adapted to the intestinal tract of humans, and it causes dysentery or shigellosis. The transmission occurs via contaminated food, fecal-oral route, feces, and flies. Most cases in the U.S. occur in children under the age of 10. For pathogenic species, we have 4 main pathogenic species that cause this. So we have Shigella dysentery, which is the most severe, Shigella flexneri, Shigella boydii, and Shigella sonii, which is the most common in the United States. An infection due to Shigella will result in the patient having diarrhea. That basically starts as water at the beginning, and then it'll switch to a stool containing mucus and blood. That's typically due to the Shiga toxins that will affect the gut and sometimes even the central nervous system. So oftentimes what you're going to get is a grossly bloody stool specimen. Sample type again is going to be stool. Sometimes it's going to be that bloody stool we mentioned earlier. Shigella and Salmonella are both typically recovered with a stool culture. We can sometimes get like that mucus from the stool, and it could be from a rectal swab as well. Again, we're going to inoculate those on differential and selective augers, so the HE and XLD plates. Shigella is non-motile. It is a lactose non-fermenter, and it does not produce H2S. So remember, H2S production should lead you to Salmonella, not Shigella. Some other Enterobacteriaceae are Edwardsella and Hafnia. So Edwardsella is motile and indole positive. It produces gas from glucose fermentation. As far as human disease, it is primarily caused by Edwardsella tarda. Hafnia, so the only species that we really care about is Hafnia alpha. It is found in drinking water, fish farm water, and intestines of fish and rabbits. Its characteristics are that it's motile, indole negative, and Vogus pascere positive at 22 degrees Celsius. It produces gas from glucose fermentation. It is urease positive, lysine and ornithine decarboxylase positive, and 90% OPNG positive, which is a late lactose fermenter.